Broadcast is now starting. All attendees are in listen-only mode.
Good morning and good afternoon. Thank you for joining Advaita Bio's open demonstration of iPathwayGuide. My name is Amanda Holden and I'm your moderator for today's demo. Advaita Bioinformatics develops bioinformatics data analysis tools with capabilities to integrate multi-omic data from a systems biology perspective. Our proprietary computational method, impact analysis, considers the type, function, position, and interaction between genes and a pathway, as opposed to other approaches that consider genes to be independent. We are a leader in the interpretation of high-throughput biomedical data, including variant interpretation, pathway analyses, disease subtype discovery, and integration of multiple data types. A few housekeeping items before I introduce our team. We will save time at the end of today's presentation for your questions. Please post your questions using the question tab at any time. Our goal is to answer your questions live. If we are unable to do that during our time together, we will follow up with you directly in email. I'd like to take a few minutes to introduce our team now, and we'll start with Justin. Justin Kennelly is our Director of Customer Success. He's the one you receive emails from, and he will be managing our polls today. Next is Advaita's life scientist, Dr. Christiana Yosef. Prior to Advaita, Dr. Yosef served as a scientist at Stanford University's Department of Pediatrics and Associate Professor of Research in the Department of Pharmacology at the University of Nevada, Reno. Dr. Yosef is also a Doctor of Veterinary Medicine. She is a Fulbright PhD Fellow in Washington University School of Medicine, St. Louis, Missouri. She completed her postdoc training in biochemistry and molecular signaling at the University of Western Ontario, Canada. Dr. Yosef led a branch of the Mountain West Clinical Translational Medicine Program in the US and has publications in over 50 peer-reviewed journals. And now, finally, I'd like to introduce today's speaker and Advaita CEO and founder, Dr. Soren Dragici. In addition to his role at Advaita, Soren is a program director in the Division of Information and Intelligence Systems of the Directorate of Computer and Informational Science and Engineering at the National Science Foundation. Dr. Dragici was elected a fellow of the Institute of Electrical and Electronics Engineers in 2022 for contributions to the analysis of high-throughput genomics and proteomics data. He was elected a fellow of Asia Pacific Artificial Intelligence Association. His work is focused on research in artificial intelligence, machine learning, and data mining techniques.
Can you hear me, Amanda? It seems like you got muted.
Hello?
We can hear you now.
Yes, we can hear you now.
Okay. You didn't hear me at all?
We lost you at some point. There was some other voice on the line. I think somebody muted everybody by mistake. So just the last part. You were talking about me.
Oh, I was talking about you. Well, let me do that again. Let me do that again really quickly so everybody can hear. And now I'd like to introduce today's speaker and Advaita CEO and founder, Dr. Soren Dragici. In addition to his role at Advaita, Soren is a program director in the Division of Informational and Intelligence Systems of the Directorate for Computer and Information Science and Engineering at the National Science Foundation. Dr. Dragici was elected a fellow of the Institute of Electrical and Electronics Engineers in 2022 for contributions to the analysis of high-throughput genomics and proteomics data. He was elected a fellow of Asia Pacific Artificial Intelligence Association. His work is focused on research in artificial intelligence, machine learning, and data mining techniques applied to bioinformatics and computational biology. He's also a professor in the Department of Computer Science, College of Engineering and Obstetrics and Gynecology, and the School of Medicine at Wayne State University. Dr. Dragici, thank you for being with us today.
Thank you so much, Amanda. So this was not a trick, okay? We didn't want you to hear this part twice, okay? It was not a deliberate trick. All right, so welcome everybody. Thank you so much for being here. As Amanda mentioned, I'm wearing a couple of different hats, and because of my affiliation with National Science Foundation, I have to state right at the beginning that any opinions, findings, and conclusions or recommendations expressed in this material are those of the authors, that is me, and do not necessarily reflect the views of the National Science Foundation. Okay, so I'm here with no connection with National Science Foundation. So basically, the first thing I would like to get an idea about who is attending this, because I will be able to tailor a little bit what we are trying to show based on your background and your level of expertise. So we have a first question. So we have a couple of questions throughout the presentation with the goal of kind of making this a little bit of a dialogue. So this is the first of them. So we can take a couple of seconds to give you a chance to answer this. And after that, we're going to show you the results. So you will also know, you'll have access to the same information that we have. So we have about 70% of the people voted. Let's wait a few more seconds. 75%.
I just closed the poll and shared the results.
Okay. I do not see that.
So the results show that 33% of people are research assistants or associates. 33% of attendees are principal investigators. 24% of attendees are postdoc or research track faculty. And the final 9% is core facility personnel.
Great. Thank you so much. I really appreciate this. Okay. So let me give you a very brief, I have one slide about the company. So basically, this is a spin-off from Wayne State. I was the director of the bioinformatics core, the Karmanos Cancer Institute, and we saw some problems with the software that was available. We're not happy. We're not getting results that made sense. So we started doing that. Then we developed these new methods. We provided these tools on our website. We started to have a lot of utilization. At some point, we had about 20,000 people, and we discovered that we couldn't maintain that as a free tool maintained by students. So that's how we started this company. So currently, we are serving 3 customer segments: individual principal investigators. I put here some examples of universities where we have people using our software. Core facilities and service providers. Again, I put here some examples of universities. And finally, some enterprise teams and pharmas. Geographically, most of our customers are in North America and Europe. That's where the biotech and research is concentrated, but we do have people as far out as Japan, Southeast Asia, New Zealand, and Australia. So currently, basically, when it comes to analyzing your data, you have 3 big choices. The first one, there's a wealth of free tools out there. And I want to bring to your attention this paper. This was published by some other people, no connection with us. What they did, they took a sample of all papers published the year before, and it's about 1,300 publications, almost 4,000 publications, and they looked at the tools that those people use, the authors. And they discovered something that was astonishing to me at the time, which is that 67%, which is like 2/3 of these 4,000 publications, reference software that captured only about 26%, only about a quarter of what was known in terms of biological processes and pathways at that time. So that's astonishing to me, because you put a lot of effort into generating data, designing your experiments, executing this. You have to obtain funding for all that research, and then one can potentially compromise the results by using tools that are outdated. So it's a good choice if you have nothing else, but there are some risks. The second big choice is in-house pipeline. So the nice thing about this is that they can do anything you want, right? Usually it's a command line, mostly is meant for and used by bioinformaticians, which is okay. But that also involves some back and forth, which can be inefficient with the PIs, right? The PIs have to ask the bioinformaticians to run this. They get the results back. The bioinformaticians don't necessarily have all the information about the experiment that was done, all the background, all the knowledge of the PI, because they have expertise about bioinformatics, not about that particular experiment, right? And the PI doesn't have the opportunity to ask specific questions. They have to go to bioinformaticians. It can be inefficient. And also it requires a lot of constant work to update and maintain these things. And the question is, why have skilled bioinformaticians do maintenance instead of tackling bigger challenges on new types of data, new technologies which are coming all the time? Finally, the third option is some kind of commercial solution. I'm not going to argue for hours, but the idea is that somebody else does the maintenance. Is meant for the life scientist, so the life scientist can interact directly with the platform, ask questions, get answers, and so on. It's going to have a graphical user interface. The disadvantage is that there is a cost that is upfront. Well, there is a cost for the in-house pipeline because you have to maintain it and have to have the people and so on. But this is an upfront cost. So it kind of has to save time and it has to provide better results to justify that cost. Okay. So given this, I want to get a quick feeling about what are you using. So we have another quick poll here. So I see that we have quite a few current users in attendance. So thank you so much for using our software and for being here. I really appreciate that. So we have like about 80% people voted.
Yes, I just closed the poll and shared the results, which show that 38% of attendees use iPathwayGuide, 37% of attendees use free online tools, 4% of attendees use free command line tools, 44% use R or Bioconductor, and 19% use IPA.
Great, thank you so much. Let's see what we can do. So I'm going to take a couple of one study here, but it involves a couple of different data sets. So basically, this is a group of colorectal cancer patients. There are 134 patients in this group, and samples have been collected prior to treatment. So these are biopsies before, when the patient is diagnosed, they take a biopsy, and before doing anything to the patient, no treatment whatsoever, they did this RNA study. Okay, so after that, the patients were given the best treatment as decided by their doctor, and that involves FOLFOX, FOLFIRI, and FOLFIRI plus bevacizumab. We're going to kind of focus on the FOLFOX and FOLFIRI because it's just one drug. And the goal here is to try to understand the resistance, right? So within each group, FOLFOX and FOLFIRI, there are a number of people who responded well, and you have the numbers here, and people who did not respond well. So we have responders and non-responders for each of them. So the kind of what we're trying to do today is try to look separately and then together at these two phenomena, right? So we are going to look at responders versus non-responders in FOLFOX and responders versus non-responders in FOLFIRI. The goals are very much similar to what you normally do in any kind of phenotype comparison. So we want to understand the differences between the two phenotypes. In particular, for one particular drug would be the differences between responders and non-responders. We want to identify the genes, biological processes, and pathways that are different between responders and non-responders for each drug. We want to understand the mechanism involved in the drug response. We would like to identify critical upstream regulators, if any. We want to identify potential combination therapy drugs. So maybe if we can, from our data, from our analysis, if we can identify a drug that could be given to the non-responders for a particular drug in order to improve their response, that would be ideal. The input is the differential expression data. So basically, this is two sets of measurements between the groups, and we have differential expression calculated between the groups for every gene. The workflow is going to be following the same question. So the first question, the input is going to be gene expression data, and the first question is going to be what are the differentially expressed genes. So here to the right, we show the tab in iPathwayGuide in which that particular question can be addressed, right? So as you're going to see, if you're not familiar, the iPathwayGuide has a number of tabs. They go from left to right, from simple to more complex. And this question about differentially expressed genes is going to be addressed on the genes tab. Then we're going to go and look for biological processes. This is in a gene ontology tab. Key pathways, biomarkers that can be used to predict the response to this particular drug. We can try to identify upstream regulators. That could be genes, could be microRNAs, could be long non-coding RNAs, and anything that may be relevant. And then maybe we can identify drugs that can be used in combination with the target drug. So I want to leave this workflow on the screen for a second and ask you, sorry about that, what is the most challenging step in this workflow from your own experience? So which one do you think this is the most challenging, or could be more than one, of course? So Justin, are we showing the poll?
The poll is now live.
The mechanism is in the lead so far. Upstream regulator. So I have so far mechanism is 40%. Upstream regulator is 21%, with about 80% of people responding. Meta-analysis of several data sets is following 15%. Also, biological processes. Okay. All right, so we are going to try to do all of these today, and let's see what you think about this. All right, is it going to be different? Is it going to be a little bit better from what you're doing? Who knows? It's a decision that you're going to have to reach on your own. I'm going to jump into the software, and we start looking at this data. Can you still see my screen?
Yes, we can.
Perfect. All right, so this is the, okay, I should start with the dashboard. I should start by logging in, of course. So we have several tools, one for variant analysis, single cell, and so on. Today, we are focusing on iPathwayGuide. So here to the left, this is the dashboard. Here to the left, we have some directories. In each directory, we have a set of analysis results, if you wish. And for each analysis result, we have the date, We have a printable report that is generated automatically. If you want to share with somebody, you just click on this and add their email. And once you do that, you click the share button and they receive a link allowing you to do exactly what I'm going to show you. But let's just go into this. So I'm opening this particular result. This is a report for this particular contrast. Actually, I have all three. I have three contrasts, three sets. I have differentially expressed genes that are found in the FOLFIRI responders versus non-responders. I have differentially expressed genes, actually all the genes measured between the FOLFOX responders and non-responders. And I also have a set of biomarkers that are coming from a live scientist, right? So from an expert. So we ask somebody with expertise in this area, look, if I'm interested in colorectal cancer, what genes should I be monitoring? What genes should I be looking at changing? And they came up with this. They used a platform annotation database, and they came up with this particular set. Okay, let's just start with the FOLFOX. So now we are going to focus on the one contrast, which is the difference between the responders and non-responders in the people treated with FOLFOX. Okay, so as you see here on the top, the tabs that I was mentioning. So from the left to right, you go simple to more complex genes. Then you have gene sets, various types of gene sets. Then you have pathways, various types of upstream regulators, diseases, and networks. Here, what we see right now is a summary. There is one such summary for each type of analysis, so there are many. So what we are looking right now is the summary for pathways. And if we look at this for a second, this is a very information-rich diagram. It shows you the pathway. The pathway is here. They have the p-values. The p-values, the scale is here. So the darker the green, the more significant the pathway. Here you have the genes, and you have the fold changes. So red is upregulated genes, blue is downregulated genes. And the chords are showing you which pathway each gene belongs to. And if we look at it, the top pathways, so currently we have 6 pathways. Let's increase that to maybe top 10 pathways. And 10 genes per pathway. And we look at them, and actually ECM receptor interaction, proteoglycans in cancer, focal adhesion, amoebiasis. This is a disease that is not particularly relevant here. So let's say I want to remove this from my graph. So I just go here and I just remove this, and I do this. The other thing that I want to do here, I want to move this a little bit to the right. So I'm going to rotate this a little bit so that the pathways are on the right. Okay. So this is a diagram, as I said, that contains a lot of information. For instance, I can very quickly see that the genes on, let's say, chemokine signaling pathways, which is this purple here, they are all downregulated, right? And so on. So you can get a lot of information from this thing. Once maybe this is a little bit too busy, protein digest, I want to just eliminate some of these pathways, and then I want to increase the font size a little bit, and then I can save it and I put it in my paper. Anyway, so basically we look at a couple of different pathways. This is the very big picture, first impression. We have a couple of big pathways. They kind of make sense. Focal adhesion, IL-17, pathways in cancer, PI3K act, and ECM receptor interactions. They make sense, but let's just go and see why they make sense. Well, let's see if we can learn a little bit more. We are going to start with the simplest question, which is what are the differentially expressed genes? So I have responders. Again, everybody has cancer here. Everybody has the same type of cancer. So we are looking at the differences between responders and non-responders before treatment. So the purpose here, if we find some differences, ideally we find some biomarkers. If a new person comes to the doctor's office, I can maybe look at that biomarker or that set of biomarkers or that pathway, and based on what I see, maybe I can say this person is going to respond or not likely to respond to this particular drug. So here I can sort by fold changes. This is the top down-regulated genes. When I click on a gene, it selects that gene. And here I have all the information about it. Among other things, I have the papers. So I would like to see if there are any papers linking this gene with cancer. So I'm going to do my my sub-selection. So here, these are all papers that are related to that particular gene, but now I'm looking at the paper that also are involving cancer. And guess what? The very first gene is the very first paper is something that actually links this particular gene to cancer metastasis. And if you look at the date, so November 24th, I could not have found a better example. This is, luck. But anyway, so basically this paper was published last week. So I want to emphasize this because some of the platforms that you may use out there, whether they are commercials or not, they update their information, let's say 2 or 3 times a year. And that means that you are basically always behind with a few months. So our references, it says here references were last updated on December 3rd, that's today. So this is not a coincidence. We are doing this on a daily basis. And this is a good experiment. You can go in your favorite platform and see if this paper that was published last week is actually available there. So this is November 24th. So anyway, so we're looking at genes. We can learn everything about this particular gene, biological processes that the gene is involved in, molecular functions, pathways that the gene appears on, drugs that are targeting this specific gene, and this is a relatively new one. So even though it's related to cancer metastasis, is not targeted currently by any drug. MicroRNA, SNPs, diseases, and so on. So this is all fine. We have a lot of information. Clearly, the genes is relevant, but there is only so much. I mean, also there are many genes that are changing. So it's very difficult to say, okay, take the first gene and say, I'm going to use this as a biomarker, right? You have to go through some clinical trials. You have to have some validation. It's very difficult to convince somebody based on just a fold change in one thing with no other explanation, no other information that this is a good biomarker. So let's just go and look at what else we can learn about these responders versus non-responders. One, we're going to start to look at gene sets. So one possibility, okay, here again, we have various choices here. We want to look at the organs that are affected. This is kind of a very, it's like a sanity check because we know that these are colon tumors, colorectal cancer tumors. So we would like to make sure that this is what we see, and indeed we see that the tissue is identified as being colon tissue. But let's just look at biological processes. So this is a very important question that initially people are paying a lot of attention, but more recently, they have started not to do this type of analysis, and I'll show you why. Let's look at this. So these are biological processes that are calculated as being significant. The calculation here is that everybody does is this enrichment. You look at the number of differential expressed genes associated with that process compared to what's expected by chance. So the problem here is, let's correct for multiple comparison, Ferroni, the most stringent correction known to humankind, and you see what the problem is. There are lots of biological processes that are still highly significant. We are looking at the top 10 right now. I want to look at the top 100. I go here, and here is the really bad news. You have more than 100 biological processes that are still significant, 10 to the minus fifth, after Bonferroni correction for multiple comparison. So this is really bad news because you have one of two options here. The first option is you can really look in detail and try to understand over 100 biological processes, right? So the first option is I'm going to look at the top, okay? Because hopefully the more important ones are going to be at the top. If you do that, let's look what you see at the top. Cell migration. Well, it somewhat makes sense, but it's so general, right? This is the gene ontology hierarchy, so general in the tree that it's really not that informative. Vasculature development, well, yeah. Blood vessel development, yeah, I know that a new tumor, a growing tumor is going to need blood vessels, and that's going to stimulate angiogenesis, but all these are very, very, very general, and also it has hundreds of genes, right? So just looking at the top is not going to help me understand much. The other choice that I have, keep in mind, I have more than 100 that are significant. The other choice, the second choice, is to say, I'm going to look at all of them. However, I'm going to kind of focus on the one that makes sense. And if you think about that, that's problematic because you are jeopardizing your chances to discover something new. If you are looking for and going to focus on the one that you recognize, you are kind of not going to be able to find something that is completely new and perhaps is supported by a lot of data, but you are just going to ignore that. And why is this happening? So this is what you get in IPA, in David, and so on. And this is the reason many people don't do this biological process analysis anymore, because you end up with so many things that are so redundant and so significant, right? So why is this happening? Well, we have this diagram here that shows you the relationship between these terms based on the number of genes that they share. So the one that currently cell migration is the top one is here, and we see that the top 5 or 6 processes, they still share 149 genes, which means that the same 149 genes are counted as evidence for all these processes. Okay, so this is not good. This is a lot of redundancy that doesn't help us. And the more you go up, the redundancy is still there, 95 genes, and so on. So in order to help with this problem, we developed a different approach. It's called high specificity pruning. And if we do that, you get much fewer things. So now you get fewer than 10 instead of over 100, with a couple of them, the top 3 being way more significant, 10 to the minus 4th compared to the others that go, this is not significant at 1% and so on. So neutrophil chemotaxis, and you see now it's much more, it's further down in the gene ontology hierarchy, so it's a much more specific biological process. The positive regulation of angiogenesis, so now is not only angiogenesis and vascular development, is a positive regulation of angiogenesis that is affected. And if we look at this, these are the genes involved in the positive regulation of angiogenesis, and most of them are downregulated. So this is much more credible, makes much more sense. And collagen fibril organization, this could be related to metastasis. And this is, look at this, this is a biological process in which all genes, differentially expressed genes in responders, are down compared to the one that are non-responders. So we have identified a process that is clearly significantly downregulated in responders compared to non-responders. Okay, so we can extract some differentially expressed genes, and now have a couple of biological processes that are important. And you see the huge difference between what the usual analysis that gives you hundreds of biological processes, and this high specificity pruning that gives you only a couple of things that are really important. Moving along, the next question is, okay, let's look at the pathways. Biological processes are just a bunch of genes. We don't really, when you look at the biological process, you have no explanation as to how they interact. A pathway is a bunch of genes, but also with a diagram that shows you the phenomena that take place on this pathway, and how these genes interact and control each other. So here, We call this a two-way evidence plot. On the horizontal axis, again, this is one of the differences between what we do and other tools that are available out there. Most other tools available out there, whether they are free or commercial, we use something called enrichment. So basically, they look at the number of genes that are falling on the pathway and compare that differentially expressed genes and compare that number with what you expect to have there just by chance. And if that number is higher, you say this is significant. That's very limited because it ignores the position and the role of the genes on the pathway. It ignores the signals, the direction, the type of the signals, and so on. However, it's still important. So we calculate this p-value, and it's on this axis. So if you were to look in IPA or in David or in some other piece of software that does some kind of pathway analysis, you have a p-value that is here on this axis. In addition to that, we have developed a mathematical model that takes into consideration all those things that I mentioned and calculates the perturbation on the pathway. So the amount of disruption on the pathway, and this is on the vertical axis. In principle, what you want to have your pathway as close to this corner as possible because it tells you you have a lot of differentially expressed genes and a lot of perturbation, okay? And so this is what you want. Once we correct for multiple comparison, this changes a little bit. And you see, you have these kind of situations here. So this colorectal cancer and this chemokine signaling pathways, if you look at the axis, this chemokine signaling pathway would not be significant if you just look at the number of differentially expressed genes, right, on this axis. And it would be significant because it is significant because of the perturbation. So this is something that you would not be able to be a false negative in IPA or in another piece of software that looks only at the enrichment. So the same thing, everything in this area of the graph, which corresponds to low abundance, fewer genes, and high perturbation, you will not be able to see, okay? You will see these things that are here, you will see these things that are here because they have high, everything that is to the right of this graph, it has significant p-values here. Let's look at the pathways. Let's look at focal adhesion. Once you select a pathway, you see the pathway diagram with the genes that are up and down. And in our software, you can look at measure fold changes or downstream effects. So let's look, for instance, at these two genes. These two genes have not been measured. Maybe they change a little bit, but their change is not big enough to be considered differentially expressed, right? But this is activated by those genes that are downregulated, and this is activated by this gene that is downregulated. So let's see what happens to these genes when I choose show me the downstream effects. So now those genes are blue because those genes are pushed down by this upstream regulation that is negative. And you can also choose to look at both of them at the same time. Once we do this, or any point, even for measured expression changes only, you can show coherent cascades. Those are the mechanisms that explain how these changes propagate to the pathway. So you can see, for instance, this part of the pathway is really not affected much, and the two entry points and these things are very much down-regulated. So this is something that, again, is unique to us to be able to show the mechanism. So you can look at measure fold changes, downstream effects, or both at the same time, and you can choose to look at the mechanism. Let's look at another pathway. Let's look at that chemokine signaling pathway. This is, these are all measured changes, right? So I have here only measured. By the way, here to the right, you have all the annotations about the pathway. You can look for drugs, microRNAs, SNPs, diseases, why the pathway is significant, and so on. I'm not going to show you all these details. I want to focus on the questions that we're trying to answer from a scientific perspective. So these are the fold changes, measure fold changes. And I say show coherent cascades. So what we see a beautiful sequence, a beautiful mechanism. So basically an edge is red if the change that is measured is consistent with the change that is implied by the pathway. So for instance, if this thing activates the next one, if this thing is down, we expect the next one to be down as well. This is indeed the case. So this is going to be a red edge. Okay. So you see this sequence of 1, 2, 3, 4, 5, 6 steps that are involving, consecutive steps in the pathway that would explain what happens on this pathway. So this would be a mechanism that would explain on this pathway, that would explain why these people, what the changes that happen in the people that make them to respond and make them different from the people who do not respond. Okay, let's move to, so we looked at biological processes. We identified a couple of biological processes. One of them was very interesting because all the genes were down. We look at pathways. We identified, and by the way, this pathway you will not see if you just look at enrichment, because it does, the p-value is just not, after the correction is not significant. You see it falls in the black area, so the p-value, the IPA will not show you this based on the numbers, right? Okay, so let's look at upstream regulators. So now we ask the question, is there a gene or some other type of upstream regulator that would explain, that would play an important role in the resistance, right? So drug resistance for 4-FOX. We are looking at 4-FOX. We look at predicted as activated. There are lots of them that are significant, but let's correct for multiple comparison with Bonferroni. Nothing is significant after that. We can change the hypothesis. We can say, okay, I want to see if there are any upstream regulators that would be predicted as inhibited. I correct with Bonferroni, it's already corrected. And I want to look for things that are genes that are as many genes downstream as possible. And the top one here is STAT3. This is kind of very interesting. It has, first of all, is not, you would not see this gene if you didn't do this type of analysis because it's not differentially expressed. So the fold change is only minus 0.48. So it doesn't mean the threshold is not considered differentially expressed. That's why it's gray in this area. However, this gene has 41 genes immediately downstream of it. And I'm going to show it to you as, let's say, subnet, no. As regulators. So basically this gene has all these genes downstream of it, and the genes downstream change in a way, this is measured change, not inferred change, right? So in a way that is consistent with the hypothesis that this gene is inhibited. For instance, this, I'm going to zoom in a little bit. So this CXCR3 is known to be repressed by STAT3 and is measured to be up. This gene is known to be activated by STAT3 and is down, and so on. So all these things, even though they change in different direction, they all support the hypothesis that this gene is inhibited. Okay. However, the gene doesn't change. So this is an opportunity for us because basically what this means, if we think a little bit about it, is that the profile, the gene expression profile of the respondents is very similar to a situation in which STAT3 would be inhibited. So this is an opportunity. The question is that comes to our mind is, can we find a drug that would actually repress STAT3, so inhibit STAT3, which would trigger all these changes immediately downstream, because those changes would make the profile of these patients more similar to the respondents, the profile of the respondents. So I go back to genes, right? So that's the question that I'm trying to answer now. Is there a drug that does this to STAT3? I'm looking here for STAT3, but remember, these are just differentially expressed, so it's not differentially expressed. So I have to show all genes first, and then look for my gene, STAT3. I select the gene, and now I look at the annotation. Is there a drug that targets STAT3? Yes, there is, napabucacin. This is very interesting. But now the question is, what does the drug do? Because if the drug activates this gene, it pushes it up, that's going to be bad. It's going to make the people respond even less. So it's really crucial to be able to find out whether this drug actually changes the gene, this STAT3, up or down. So we do a search for this particular drug. This is going to take a few seconds. And the question is, can we find some information about what specifically this particular drug does to this gene? If there are, by the way, if there are any questions, there is a section you can type in the question there. So even if I don't get to it right now, we'll try to go through all those questions at the end. I hope I spelled it correctly. Okay. STAT3 is there. Napabucacin. Okay. Effect of napabucacin and doxorubicin. Now, napabucacin inhibits proliferation by regulating GSTAT. So bingo. This is exactly what I was hoping to see. That actually this particular drug inhibits STAT3. So this is a drug that now I can consider for a potential combination treatment, right? Because I know that by looking at the gene expression changes, I know that the respondents behave in such a way that the STAT3 is down. So if I give a STAT3 inhibitor to the non-responders, I will make those changes and bring the profile of the non-responders closer to the profile of the responders. So this is another very useful piece of information. I would say this would be absolutely crucial. Okay, let's go to the network analysis. The network analysis is an opportunity that we created for the users of our platform to actually ask specific questions. In most other platforms, you put your data in and then you look at the results and you just you see what you see, right? You can self-select and release some things maybe, but you look what you can pick and choose what you want to talk about, but you really don't have the opportunity to ask specific questions. So here you have that opportunity. So let me show you an example. So I want to go to biological processes. I select about, let's say I select smallest common denominator. This is another approach that we developed. Let's select positive regulation of cell motility. Let's say this is a biological process. It has to be related to the migration of cancerous cells, right? So the ability to form a metastasis, this is kind of important here, right? And so I want all the genes involved in that. There are 98 such genes. Also, I want to focus on the genes that are present in the cell membrane because I'm looking at something that I would like to be able to drug, maybe to find the small molecule. So I'm selecting the plasma membrane. So I'm selecting the genes that are involved in that particular biological process, and they are located in the plasma membrane. And also, I want to focus on the genes that are also involved in the focal adhesion. So I selected my question, right? These are the genes. This is how they are changing. And I say, build a network that shows me how these genes interact. And here it is. So this, and I can look at this in all sorts of interesting ways. I can look for subnetworks. I can look for mechanisms. I can optimize a little bit the layout. I can look for hub genes. I can look for gatekeepers. I can look for interactions, and so on. And here I can do the same kind of analysis, the same kind of searches. I have here the types of interactions that are involved here. They are color coded, and they have the letter that also tells you the type of interaction. And let's say that okay, now I want to see what would this how would this gene set involve a particular biological process, affect a particular biological process. So I go here and I select to search for biological processes. And I go here, let's say I'm interested in cell motility. So I want to see if these things are related, or want to see if this would impact at all or affect at all the metastasis phenomenon. So I built a network, and this added this particular biological process, and now I see specifically which of these genes affect this biological process. Now I can get a step further, and I say, okay, what would happen if I were to give these people a certain drug, a different drug? Let's say I go to I go to chemicals, and let's say I'm considering tamoxifen. And the question is, which of these genes would be affected by tamoxifen, and would that propagate to this any impact this particular biological biological process? And here is the answer. So tamoxifen would act directly on these three genes, and these three genes, through a number of other genes, some directly, some through some other genes, would affect this cell motility. Let's see that this is interesting. Maybe I want to write a paper about this. Let's say, what is the evidence for this? So let's say, what is the evidence for tamoxifen actually impacting EGFR? I click on that edge, I go here, it actually shows me the paper. In this case, there is only one paper. This is the paper, and this is the evidence, basically. So I can essentially go to right to the source of that particular information. Let's say that I want to understand, okay, what is the evidence for this binding between EGFR and PIK3R1. Again, this time I have lots of papers, so maybe this is more better studied. Various types of experiment is affinity capture Western, proximity label, affinity capture MS, and so on, protein peptide, and so on. So you have this thing at our fingertip. All right, let's take a step back. So we looked at the particular one particular drug, which was Folfox, and we answered those questions: what are the biological processes, what are the pathways, what are the mechanisms, and so on. But now I would like to combine different analyses, different experiments, by looking at the same time at the Folfox resistance and FOLFIRI resistance. So now the goal is to understand whether the two phenomena have similar or different mechanisms. I will ideally identify biomarkers. I will try to identify biomarkers that can use to predict response for one, the other, or both. And maybe I can identify other drugs that can be used in a combination. The input is exactly the same. So let's go back to the software and try to answer this question now. So instead of looking at one particular contrast, I'm going to look at the meta-analysis now. So in the meta-analysis, I have my three sets. The pink is the manually curated set of genes, no fold changes. The blue is the one that responders versus non-responders for Folfox, and this is responders versus non-responders for FOLFIRI. Ideally, I would like some genes that would allow me to identify whether somebody responds to Folfox. I should give them Folfox and FOLFIRI and choose the treatment. Okay, so so I would like to focus on these 352 genes, maybe 352 plus 5. Well, but I would like a gene that would go up if somebody responds to Folfox, and down if somebody responds to FOLFIRI. So because I want to distinguish between those, right? Unfortunately, this Venn diagram doesn't allow me to do that. So I can select those things, fine, I have them, and now I can look by one, which is going to be painful because I have 352 of them. But we have this other tool here. It's called offset plot. And here, I say, this is the same thing. So those 352 genes that are there are here. Those are in common between FOLFIRI responders versus non-responders and Folfox. So it's another way of looking at the intersection. And these are exactly the same areas that you see in the Venn diagram. However, here, I can say, I'm interested in the direction. If I do this on the Venn diagram, I'm going to end up with so many areas that I'm going to get lost. But here, I say, I'm interested in those genes that are positive in Folfox and in the FOLFIRI responders and negative in the FOLFOX responders, and redraw showing only those. And I select only those that are listed in plus in FOLFIRI responders and minus in FOLFOX responders. And I select just the ones that I want, and now I can sort them by log-fold change, whichever way I want to go, and I start to look at them. And for instance, this call 1-2A1 goes up 1.3-fold in FOLFIRI responders versus non-responders, and goes down in FOLFOX responders. So here I have basically a list of genes that any one of them could be potentially be used, of course, after validation and so on, to direct the treatment. So this is something that is going to tell me whether a person is going to respond to one versus the other. This is all good, but again, looking at individual genes is not very reassuring. So let's look at biological processes. We can do the same thing on biological processes. So here, I select my high specificity pruning, and I select, I order by FOLFIRI responders, and the very top one is collagen fibril organization. So now I have a whole biological process that is very significant in both. So this is a process that is, again, very different between responders and non-responders in both FOLFOX and FOLFIRI. And here I can go and I can pick any one of these genes that where the gene goes in opposite direction, maybe the one that goes with more changes in both ways. So that's another way to do it. But now I have the reassurance that I know exactly what biological process these genes are involved in and what I'm monitoring. Okay. Let's go to pathways, because that's where the most interesting things are going to be. So now I can look this, I can sort it by FOLFOX or FOLFIRI, and I can ask myself what happens with one pathway that is important in, let's say, FOLFOX. What happened to the first pathway here in FOLFIRI responders? So just by mousing over, you see what happens. For instance, this is an important, very important pathway in FOLFOX, very important, it's number 4 in FOLFIRI, but it was not, my expert and my knowledge base did not suggest a lot of genes to monitor on this pathway. So this is something that I would have missed again. So click, I select, let's say I select one of these pathways. Let's go Bonferroni. Let's go here, and let's say look at focal adhesion. I look at the pathway diagram, and now on the pathway diagram, I can choose to look at my biomarkers chosen by the life scientist, and you see it monitors very well this sequence of events, but nothing in this part. And I want to see what happens in FOLFIRI, what happens in FOLFOX. Maybe I want to see the mechanism as well. So this is what happens in FOLFIRI. Let's just do the downstream effect and the combined. So this is what happens in the FOLFIRI responders. This is what happens in the FOLFOX. So basically, you see this entire part of the pathway is downregulated in the FOLFOX responders, and it's, if I go to FOLFIRI, this is upregulated. All these genes are upregulated in the FOLFIRI. So this is, again, I identify the pathway that is different between those, and the specific area or mechanism of the pathway that makes the difference, and is very different between the two drugs. So now let's go back to the question. I would like another combination therapy. I found one for the FOLFOX. Let's see if I can find something from FOLFIRI. So I'm going to go back to the FOLFIRI data, and we have another capability, which is to look at directly at the chemicals. Okay, so we found that looking at STAT3 upstream regulators and then finding the drug that does that. But we can also look directly at the chemicals. The chemicals could be drugs, and I'm clicking here to do that. And I correct for multiple comparison. And the top thing is dasatinib. So this is, again, I have 411 genes impacted by this drug. 109 of them in responders behave as if the dasatinib is present. So this is, again, if I give dasatinib to non-responders, I will expect these 109 genes to change in the appropriate direction and make their change profile, the expression profile, more similar to the responders. So this is another drug that I could use as a combination therapy. I'm going to go back to the PowerPoint, and let's just summarize. So basically, we had a couple of questions that we wanted to answer. The first one, what are the biological processes that are different between responders and non-responders? We started with fall fox. We found a couple of ones that are interesting: collagen fiber reorganization, neutrophil chemotaxis, positive regulation of angiogenesis. The next question was, what are the mechanisms? And we found a very nice mechanism on the chemokine signaling pathway that involves chemokine receptors, GNAI1, FGR, and then PI3K. Another question was, are there any critical upstream regulators? Yes, we found STAT3. It was right at the top here, and that helped us identify a drug later. Are there any biomarkers that can predict response? Was the next question, and the answer is yes. Look at these two pathways; it's like night and day. To the left, you see the respondents for folks. To the right, the respondents for porphyrin, and you can see that by looking at this pathway, now you have a new patient. You can take a sample, do a biopsy, and then look at the genes on this pathway. And if it's more similar to the left, you know what drug to give them. If it's more similar to the right, you give them the other drug. Another question, can we find any drugs that are likely to increase the response to this? And yes, for folks, we found naprobucacin. For porphyrin, we found dasatinib. And here's a paper that actually showed that this inhibits STAT3, which is exactly what we wanted. Dasatinib was right at the top in the chemical drug analysis for the other one. So, and finally, the last question, do these resistance phenomena for the two drugs, do they have similar or different mechanisms? And what we found was that the mechanisms are different, but they involve the same pathway. And I showed you in the software, this is a different pathway that you can see. This entire part here is down-regulated, is a very nice mechanism. Again, this is nothing inferred, this is measured. And this does not happen in the other one, and these genes are all up-regulated, which does not happen here. So I will stop here, and we'll be happy to, I think we have another question for you, and we'll be happy to take questions in the meantime.
Great, thank you, Soren. So the first question we have is, I'm unable to add the coherent cascades to my pathways. Is this something that will not show up if I do not have downstream effects appearing?
Yes, there are situations in which this doesn't happen. So one situation is a pathway. Sometimes you have biochemical pathways that do not contain any signals between genes, like gene regulation. So if it's a metabolic pathway, for instance, that has reactions, not signals between genes. So metabolic pathways, you cannot do that. Also, that is not available if there are no coherent chains of perturbation propagation. So that if there are no sequence, there is no sequence of successive steps in which the things that are consistent, the measure values are consistent with what the pathway says. In that case, that thing will not be available. Would be grayed out.
The next question, I believe, is related to the model you used. Why do we get differentially expressed genes related to colorectal cancer if in this sample we are comparing two sets of colorectal cancer?
That is a very good question. So we do not get necessarily, so if you look at the, let's say, the meta-analysis here, let's go to the Venn diagram. It's easier. There are that's true to some degree. So you have these things are in common, the 11 plus 5 plus 9, but the 77 genes are genes that are changing, that this human expert said that are changing in colorectal cancer, should be monitored, and apparently they change in both responders and non-responders because everybody has colorectal cancer. So these are the genes that are kind of not different between responders and non-responders. These are the genes that are different between responders and non-responders.
Okay. Do you have software for kinase prediction from the phosphoproteomic data?
Yes, that's a really good question. So we try to be agnostic on the type of data. So basically, you can analyze in our platform. In this platform, we can analyze any kind of data that can be mapped on protein, effect size, and some p-values. And the p-values are optional. The effect size is optional too. So whether it's phosphorylation, you can still map it on genes. You can put the phosphorylation level and the p-value, and you can analyze that. So it's not, and it's going to predict. It's going to do all the analyses that I have shown. Well, I haven't shown all of them, but it's going to do everything that can be done with a set of genes, including predicting upstream regulators, including showing where the pathways and so on and so forth. So it's not as specific as your question was aiming for, but that kind of data can be analyzed, and you will have predictions, including the kinases.
Okay. Do you offer a free trial?
Yeah, thank you for that question. The answer is yes. You can create an account, and that would give you access to the platform, and it will also provide access to a number of data sets that would allow you to do everything that I have done right now. So you can use the platform, full capabilities, full features, but for those data sets only. You cannot really analyze a new data set with a free trial, and that is because we are using AWS, which is the Amazon platform, and we get a bill and incur costs every time a new data set is analyzed. So for that reason, we don't provide that for free. But you can have free access to the platform, and you can explore it at your heart's desire.
Is there an expiration for free access to the platform?
No. So you have an account. You create an account. You have access to the free data sets, the demo data sets that are included there for as long as you have the account. At some point, I mean, I don't know, if you come 20 years later, if you haven't touched me in 20 years, we may have deleted that account, but there is no set date. I mean, it's not that you can use it for 2 weeks or something like that, right? So you have the time to explore it. So the other opportunity that you have, you can, if you really feel that you want to compare this with a different platform or something, we can offer a limited trial, like a 90-day trial, or even you can pay for one analysis. But that's not something that is let's say cost effective. So that makes sense for one analysis or two analyses to make a comparison, but that's not how we recommend. The idea is that, as you see, you send your data to be analyzed once, but then you come and do all this analysis, and you can ask those questions and do those, explore your data and construct those mechanisms and look at the gene networks and so on. So and you want to do that repeatedly. So that's the value that we provide. It's not just the one-time analysis. As I said, this is a little bit of a different paradigm. Instead of submitting the data, getting the results, I'm done with the analysis. This is a platform that allows you to really truly understand your data and ask questions about it and evolve your knowledge. So the value is in once you analyze your data, you got the results, but then you do all these explorations and you do all this correction. Everything that I've done here is after the so-called analysis, right? Which was done at the beginning.
Are we allowed to define our own gene sets or pathways and run the analysis?
Well, that's a really interesting question, and that's the reason I included this. Not as a pathway, but you see, you can upload it as a different input, exactly the way I did it with this particular set. So this is my set of biomarkers, which will be your set of interesting genes for whatever reason, and then you can look at what are the pathways that these genes impact and so on. And also you can look at meta-analysis between measured changes. So exactly this, I think this is a good example. So this would be like how is this particular, let's say this is my custom pathway, right? By the way, you can actually create a custom pathway beyond KEGG. So you can go on a pathway here, and let's say I want to look at, I want to create a pathway that involves these genes, but it's not limited to the pathway topology that KEGG has, right? Because I think this is limited. So what I can do, I can go here, and here down is going to create that pathway based on my measured changes as well as all the known annotations. And here again, you can subnet and you can customize this. For instance, okay, these are not related to anybody. I'm going to hide the isolated nodes. So I'm going to let it finish. This is my mechanism. I'm going to hide the isolated nodes. And then you can look at regulators, and you can look at the evidence for it, and let's say this thing here, let's say, oh, this doesn't apply to my thing. I'm going to hide it, and that disappears. So you can customize the network and create kind of custom pathways in this way.
Great. You mentioned competitor a few times during your presentation. Who is the competitor, and do they offer packages better than yours?
The competitor is whatever you use right now to do this type of analysis. That's our competitor, okay? So if you use Excel, that's our competitor. I think that we're going to provide much more information than Excel. So this is not about a competition. This is about, you know, can we help you do a better job at understanding your data, okay? So some people use, as I mentioned, some people use free tools. Some people use R code that they wrote themselves, or they use from Bioconductor and so on. So all those are alternatives. Okay, so we think that our goal here was to create a platform that is really user friendly. It can be used by a life scientist to ask specific questions without waiting for other people, without using command line and so on and so forth. So that was our goal. And also we try to develop these methods that are going beyond the limitations of the existing methods used by other packages.
Can we analyze microbial metatranscriptomics data?
So if the question is, can I analyze gene expressions coming from a meta-analysis of a microbial sample that involves God knows what species? The answer is no. We are supporting human, mouse, and rat, and we have people who used our platform very successfully with many other organisms. For instance, mackerel and things like that. So not model organisms and nothing close. And the way you do this, you map those genes to one of the 3 model organisms, and then you do the analysis. Okay, but we do not. This is not meant. For instance, you can use the effect of a microbial species as it affects a human, mouse, or rat. But to analyze microbial genes themselves, we do not support that at this time.
This seems like a pretty comprehensive platform. How long does it take the average researcher to learn how to use the tool?
Great question. So what we do, we provide trainings. So we provide an initial training session, which is about an hour and a half, 2 hours. And that takes it from the very beginning to the end, loading data set, all sorts of choices and so on. And that gets most people very comfortable with the platform. As you see, we also have these little camera things here. If you click on that, you have access to a 1 to 2 minute video that tells you exactly what this part does and so on and so forth. So we have this throughout the application. So people get very comfortable very quickly. Then they start using it, and then 3-4 weeks later, they can become very sophisticated, and they can have additional questions. At that time, they can contact us, and we can offer additional kind of advanced training. That can happen anytime. If it takes you a week to do that, we do it a week later. If it takes you a month to do that, we do it a month later. So basically, it's up to you. And this is all unlimited and at no additional cost. We also provide support with everything that comes from our analysis. If you want to write papers, you have to respond to reviewers. You have some question about an analysis part. You can share that with us, and we can provide a draft response and help you address it properly. Whatever it is, a grant, a paper, whatever it is, we can help you with it.
How much does it cost to share analyses with our collaborators?
Oh, this is actually free. This is another major difference from other commercial platforms. In other platforms, if you want to share, the recipient has to have a license as well. In our platform, the recipient doesn't have to have a license. So if you have a license and you have collaborators at the other end of the country or the world for that matter, that you like to team up with, you can share the result with them. They have access to everything. They can go, for instance, and ask specific questions here and create networks and so on. And there is no additional cost.
Okay. And I believe this is our final question. Can you share how the pricing model works?
Thank you for that question. So we strive to do is to meet everybody's needs in terms of pricing as well. As I said, I have to get grants myself. I was the bioinformatics core director. So the way we decided to do that is we can offer customized pricing, and there are two variables. One variable is the volume of data that you expect to analyze in 1 year, right? It's a 1-year license. And the second variable is the number of seats. So if you have, let's say, 3 people in your lab, and you expect to do about fewer than 100 analyses, then we can configure a license that takes those two numbers into consideration and comes up with the price. So basically, a big lab is going to, it's unfair to ask one PI with one postdoc to pay the same amount of money that a lab with 20 postdoc would have, or a commercial company, right? So we can customize a license based on your needs, based on those two variables. So if you can contact us, we'd be happy to give you more details and try to figure out something that will work for you.
Wonderful. Well, I believe those are all the questions we have, and I believe we've answered all of them live. For those of you who we did not provide a lot of details, Justin will be following up with you directly. So at this point, I'd like to thank all of our attendees for joining us and staying with us for the entire hour and 15 minutes. And Soren, thank you so much for your presentation. And we wish everyone a great rest of their day or evening. Until our next webinar, we wish you well.
Thank you so much, everybody. It's been a pleasure.
I'm going to end the webinar now.
Hold on. Bruni has a question. Bruni, if you are still here, please ask your question. Or maybe not. She just logged out. Okay.
Okay. I'm going to end the webinar.
All right. Thank you.
Thank you.